Hi there. I thought I would help you continue to make progress on your bacterial transformation lab since it's a snow day and we won't have the lab report work session today. So this is where all of you should be at this point. This lab, um, this data or a model of a data table is on page 43 in your lab packet. And I'm actually just going to be walking you through your lab packet right now. So that's the only thing that you need in front of you. And this is where all of you should be, except those of you that did the lab yesterday. You're not quite here yet. So each lab group should have a group data table. And each data table should have the following things in it. You want to have, first of all, this data table doesn't show it, but you should have a precise title at the top. And then you should have your treatments. It doesn't have to be set up exactly like this. This is just one way of setting it up. One thing that this data table doesn't have in it is your pre-transformed data. So that should definitely also be in here. These are the treatments, but you definitely need your wild type bacteria that you observed before the transformation in order to make any statements about change. You need to know where you started from. So pre-transformation information should go in here as well. And then this is all your post-transformation. You have your two treatments that you gave your engineered plasmid, the plus P-low, and then the two control plates that you didn't. If you use any abbreviations, they just have written here, LB is a standard abbreviation, but AMP and ERA, if you're going to use abbreviations, then you need to clarify somewhere what those abbreviations mean in order to use abbreviations so the reader can clearly interpret what you mean in your data tables. They show here a space to add drawings. I would rather that you please include photographs in your data table. It's just a more precise way of including a visual record of what you observed. And then for your observations, I'd like for you to have your two different types of data. Please include both qualitative data, so what your treatments look like, what the bacteria look like, the color, maybe the shape of the colonies, whether it was a lawn. A lawn is the term that's used for when all of the cells are growing closely together. Or maybe if there's any altitude, so texture to the colony, so you're describing what your bacteria look like. And then also you want to include any quantitative data. So the number of colonies growing on each of these. A colony, remember, is just a little circle of bacteria. The actual physical size of those, so measure them. That's why you had a ruler. Maybe use your ruler to measure how much space is in between, so the spacing or the pattern in between the colonies. You want to talk all about that. Okay, so that's what goes in your data tables. And an example of a data table is on page 43 in your lab packet. After you're done with your group data, each individual written lab report, after the group shares all the information, should look a little bit different. So you should tailor your data table to be specific to your written lab. So think about those finer adjustments that you'd make to make it your own. Okay, so the next step once you have that data is analyzing your results. You have to write the next two sections of your lab report are your conclusion first, and then your discussion section. So let's start by talking about your conclusion. In order to write your conclusion, you have to understand whether or not you supported your hypothesis. Let's take the example that you decided that your hypothesis that you stated in your introduction was something about the universality of DNA. So you're talking about how all cells speak the same language. So in order to determine, the first sentence of your conclusion is whether or not that hypothesis was supported or not. And so you have to figure out what your data and observations, what your data table means in order to do that. And I think a good way of doing that is answering the analysis question. So before you write your conclusion and before you do your analysis, or pardon me, your discussion, I think a good next step would be to complete the questions on page 44 in your lab report. And then there's 4 questions on page 45. And then there are 3 questions on page 46. So there's 3 pages of analysis questions. So I would do that next. Now, all of these questions are just getting at the basic claims and the purpose of your investigation. So, for example, if you're let's talk first about your conclusion. If your conclusion, I mean, if your hypothesis is something about the universal DNA as the universal language, then you need to talk about how a prokaryotic cell expressed a eukaryotic gene, right? And that's what these questions ask. So this is talking about originally what the traits were, and then after the transformation, what the new traits are. That's what's on page 44. On page 45, it's making sure that the glowing is, you're justifying the glowing as evidence for a new phenotype and that something else isn't glowing. So you're making sure that your evidence is, you're justifying your evidence that the transformation was successful. So those things are important when you write your conclusion. You're wanting to provide evidence, your data as evidence to support that universality of DNA. You need to make sure that you are, in fact, looking at a new gene that's being expressed in a cell that didn't have that gene before, if you're looking at one cell speaking another cell's language. And then your conclusion would have a third sentence that would justify that data as evidence. And you can see in your analysis questions, there's questions that help you get at that. So for example, number 4 here, it says, from the results that you obtained, how could you prove, provide evidence that the changes that occurred were due to the procedure that you performed? So there is making sure that you understand that you have to justify your evidence. Okay, so after you write your conclusion, then there's more questions here that help you look at some of the other claims. Remember, that's your discussion section. In your discussion section, you're taking that your original hypothesis, that's how you start. Again, it sort of comes from your conclusion. A little bit of the beginning of your discussion restates your conclusion. And then after that, you take 3 or 4 more claims from your discussion and you're providing additional evidence. So you want to explore, look at this data table. You want to explore more of the data. You want to take this evidence here and see if you can use this evidence of these treatments, that all of this data here in your data table. You want to see if you can use that data to support more of the claims in your introduction. Like, for example, if you look at the questions on page 46, these questions get at the interaction of genes and the environment. So you likely talked in your introduction about the importance of cells responding to the environment. Why does that matter? Why do cells have to respond to the environment, and what would be important about that? And then you can make the claim that they do. Maybe you said something about that in your introduction. And then you can provide evidence to support your claim looking at your data table. So that's what your discussion is. That's the first part of your discussion, the bulk of your discussion. The second part of your discussion is your error analysis. So I'm going to start another video to talk about your error analysis.